Inno Podcast, un proyecto audiovisual de Inno Campus Explora.
Hello and welcome to another episode of Inno Podcast, the podcast of the Inno Campus Explora project. I am Marta Serrano López, a student in biochemistry and biomedical sciences, and today I will be interviewing Ronaldo Ichiyama from the University of Leeds. His areas of expertise include motor control, locomotion, neurodegenerative changes following a spinal cord injury, neurodegeneration, and structural and functional plasticity in the spinal cord. Welcome, Professor. It is a pleasure for us to have you here at the podcast.
Thank you very much. Thank you very much for the invitation. It's a pleasure.
Could you explain shortly for us what your trajectory has been so far?
My trajectory. So it's a very long one. So how much time do we have? So I was born in Brazil. So that's why my name is Ronaldo. I did my undergraduate there. And then I moved to the USA to do a master's degree and then a PhD and a postdoc. So I actually moved from sport and exercise sciences, in the US it's called kinesiology, into neuroscience. So my trajectory into the neurosciences were a little bit late when I decided to study how the brain controls movement and how the brain is actually affected by movement. So for example, when you exercise, what happens to your brain when you exercise and when you don't exercise. For my postdoctoral career, then when I finished my PhD, I moved to Los Angeles to work at UCLA, so the University of California in Los Angeles. And there I started working specifically in spinal cord injuries, trying to help people regain function again. So ever since my postdoc, I have been working on spinal cord injuries, and now it's been quite a long time. It's more than 20 years now in this field.
That's really interesting. Actually, we asked this question because as students, we are really interested in how people reach the position they're in, how they may change fields and all that. And sometimes it's quite scary to think, what am I gonna do with like my future and what am I gonna choose? So knowing that is kind of reassuring for us because we know we can change our mind and like go to other areas of study and maybe what we studied before help us in that other area. Isn't that right?
Absolutely. So I missed the part there. I was actually a gymnast and I was a gymnastics coach before. And look at me. I'm a professor of movement control and neuroscience right now. So it's a different journey. So yes, your life would just, opportunities will show up to you and you just open the doors where you feel that it's more interesting to you. So, you know, you never know what doors are going to open.
Oh my, I wasn't expecting that. Anyway, while Professor Ichiyama has several areas of study, as we've seen, today we will focus on spinal cord injury and regeneration. In order to understand this subject better, we would like you to start with a brief explanation of what neural regeneration consists of and the mechanism involved in the after spinal cord injury.
Yes. So spinal cord injuries is one of those conditions that a lot of us know or have seen it. We have experienced it either, you probably know somebody that has had it, and you always associate spinal cord injuries with people sitting on a wheelchair. So that's not always the case. So sometimes they're walking about and you don't even know that they have a spinal cord injury. So the major problem with a spinal cord injury is that any injury is not just to the spinal cord, any injury to the central nervous system right now is more or less permanent. There's not much we can do about it. So it's different from, for example, if you cut your skin, if you break a bone, even if you take part of your liver out, part of your body regenerate. You cut your nails, you cut your hair, it grows, your skin, right, scars, and everything is more or less fine. But when you do that to the central nervous system, it doesn't happen. And for a long time, we didn't know why that was. And it's quite interesting that since the late 80s, 90s, we discovered that there are several problems. And one of the problems that we didn't even understand was that we produce certain substances, some molecules that actually prevent you from repairing, which is unheard of in other areas. It was unheard of in other areas. So usually when you have a problem, the body gathers its energy and gathers all of its systems and try to repair it, so you can continue walking and you can continue eating and so forth. But for some reason, our central nervous system doesn't have that capacity. So the discovery of those inhibitory molecules, we call them, was a very big step into our understanding of why it doesn't repair. It was very interesting because at the same time that you have this increase in inhibition, what we call things that prevent you from getting better, there's also a decrease in the things that make you better. So if you're in biomedical sciences, you know what growth factors are.
Yeah.
So those are down regulated, so you have less production of them at the same time that the inhibition ones are going up. So it's very counterintuitive. Why are we doing this? We're not even trying to repair the central nervous system. So that's the problem with stroke or head trauma or any of those types of insults to the central nervous system. So a major obstacle in spinal cord injury today is try to find that holy grail of how can we actually make something regenerate for real. So reconnect from the part that was cut or damaged to the parts that are not damaged.
I see. We will talk a little bit about that.
Okay.
Because we have a question actually about regeneration of axons. But if you allow us to start with, we would like to talk a little bit about what my colleague coined as scientific gossip. Is there any specific project in which you are involved that you believe could have a really big impact in our community in the future?
For spinal cord injury specifically. So I have been involved for a very long time now on the use of what we call, it's a jargon term, that we call it neuromodulation. So it means that we can use artificial things, so not things that are within the central nervous system or your body, to actually help the system do something useful. So in my case, I use electricity. So I use electrical stimulation of the spinal cord. And from the beginning of my studies, we studied that in animals first. We saw really good results that I could actually make a completely paralyzed animal walk again with this neuromodulation, so this electrical stimulation. And that study has progressed so far that now it's been applied into humans already, and it's been translated by several different groups around the world. And it's quite exciting to see that something that you started in the lab with animals is actually having clinical results. It's not widespread available yet. It's still experimentally used in the clinic. So it's clinical trial stage, but it's having really good results. So I'm very hopeful that that kind of line of research and application, intervention can actually help people. That's not the cure for spinal cord injury. It's helping them. So if you couldn't walk before and you can walk now, you couldn't stand before, you can stand now. That's a step forward, but it's not the cure yet. So that's one of the things that we are pursuing with other lines of research.
So it's not targeting the cause, but at the end of the day, it's helping those people.
Yes.
So I remember that in one of your classes, I saw this video of a mouse that couldn't walk, and then with, I think it was like epidural stimulation, they just started walking out all of a sudden, and it was amazing, and I was amazed by it. And just the thought that someone that cannot like move the lower limbs at all and can walk thanks to that is just amazing.
So unfortunately, like I mentioned just now, it's not widespread available yet. But the people that have been participating in these studies have gained a lot of function. It's quite incredible, actually. The problem is that it involves a lot of effort to get to that point. So it doesn't just happen like that. So it's a little bit complicated.
It's just normal. The central nervous system is just... even though of idea of a big discovery or milestone in science, as we've been talking about, is restricted to geniuses by themselves in a lab and they're like brilliant ideas. This is not reality. In reality, it is important to recognize the role that research teams as a whole play in this. With such a range of areas of study, including the ones that yourself have been through, I assume different kinds of experts are needed to look at these topics in depth. Are students and researchers from different scientific backgrounds present in your lab? And if so, which of these scientific areas could be highlighted?
Yeah, so that's a very good point, and that's something that people that are not within science, more or less in science, they don't realize how much effort it actually takes. In the case of spinal cord injuries, if you think about it, when the spinal cord is hit in a car accident or fall or something like that, it's not just the fact that there's trauma to the central nervous system and you're just gonna put a little magic pill and it's finally just gonna regenerate again. It's a lot more complicated than that. It involves so many systems. So even the biological systems, we have to work with not just neuroscientists, we have to work with immunologists, cardiovascular specialists, exercise rehabilitation scientists, but also there's a lot of emphasis in nanomaterials, for example, biomaterials, so engineering, mechanical engineering, electrical engineering. I just mentioned my epidural stimulation studies. That's all done in combination with electrical engineers, computer science, modeling. There's a whole group of people. You can think about physics, chemistry, biology, all merging together to try to understand how best to help. So yes, even in my lab, I have students from different backgrounds, depending on the project and depending on the part of the project. So if you see major publications in the field of biological sciences in general, not just spinal cord injuries, you see that the list of authors is growing and growing. It's never one or two. It's a lot more than that because the effort is really astonishing that you need. So, and it's a good thing because working in the team pushes you, each one comes with different set of expertise, and you together you can make something a lot bigger than you or one or two people. I'll tell you something, just an aside from this here. When I was still doing my postdoctoral fellowship in the USA, I worked with the Christopher Reeve Foundation. So Christopher Reeve, for some of you that might not know him, he was a very famous actor in the US that got a very severe spinal cord injury falling from a horse. And he's the actor that played Superman in the 80s movies, I can remember. So after the accident, he started his own foundation, the Christopher Reeve Foundation for Paralysis. And within that, one of the mission was to gather a group of leading scientists from across the world to work together to find something, to find some way of improving the situation. So back then already, it was already established that one person cannot do it. There are too many variables and there's not one specific specialist that can solve all of the problems. You need them all at the same time.
Yeah, but isn't that wonderful?
It is, and it's the idea of what science should be, right? That we share and we discover things together and like, oh, wait a minute, right? It's really exciting.
Our whole project, the Campus Explorer one, is focused on that. So we always have both students and teachers from different areas that are way, way different. For example, in my group right now, I'm with other people from biomedical sciences, but also with someone from physics and someone that is studying engineering, like I think informatic engineering. So it's really interesting to see what the points of view are regarding all these topics.
Even areas that you don't think about, like social sciences, arts, and there's a lot that can be exchanged between the fields.
That's completely true. I cannot say anything else. A notable branch of your research is based on the hypothesis that the combination of locomotor training with different neural regeneration strategies are the keys to successful neural regeneration after injury. What led you to these conclusions? And what are the benefits compared to employing both strategies separately?
So, one of my early emphasis in my research in spinal cord injuries was just to understand how physical rehabilitation, so exercise, physiotherapy, could actually improve movement. If it improves, how is it doing it? One of the things that I noticed very, very early on, you can improve, but it's always limited. It's not enough. Nobody has been able to just get up and start walking again with physiotherapy only. If it's a very severe lesion, sorry. If it's a moderate lesion that you're almost about to walk, rehabilitation absolutely will help you, and you'll probably walk again just with rehabilitation. But it's a very severe lesion that never happens. So in those severe cases, which is quite a bit of the cases, just rehabilitation wasn't enough. So we needed to actually help the system try to regenerate or try to do something that it's not able to do on its own. So I mentioned the inhibitory molecules, so the ones that are preventing regeneration from happening. So what if you just stop them? If you block them, something happens? Yes. When you do that alone, again, you see some improvement, but it's not complete. So today, so you can increase, for example, you can block the inhibitors, you can increase the enhancers, so the growth factors, you can give them growth factors, you can give them drugs that can help this or that neurotransmitter, but it never is enough. And one of the problems is, okay, so if you have a little bit of regeneration, which is actually happening now, why are we not seeing this recovery of function that we should expect? It's because one of the reasons, and one of the major reasons, you see axons regenerating beyond the lesion, but how does it find the actual target that it had lost in the first place? That's a really difficult question to answer, and that's a really difficult task to complete in biological sciences. So in development, when you're a fetus or a baby, it happens more or less naturally. There are programs that guide all of these connections that should be happening and should not be happening, and it's programmed within your genetic makeup, but it's also dependent on activity. So if you don't move, it doesn't know that your muscles are there. So you, as you're developing, so it finds this target. So it's a little bit similar in the sense after an injury. So you have something growing, but you need to have the targets posted for you to find the target. Rehabilitation can do some of it. So exercise can do some of it, because with exercise we know that some factors are produced and things that are growing the system like that environment. So that kind of combination is probably the best thing that we have right now in regards to actual regeneration with functional regeneration. So those two things are quite important. Just growing things doesn't really mean that you're going to regain function. And sometimes you don't. Sometimes you get pain, for example.
Oh,
right? That things grow and you get abnormal pain. Okay, that's really not what we want. Right?
It's really interesting because the other day one of my teachers in developmental biology, we had discussed on regeneration, and she said regeneration sometimes is kind of, in some animals and in some systems, is kind of like development all over again in a way. So I found it really interesting what you said regarding that. I know it's not exactly the same, but I would like to link my next question with what you said about like axons. With the problem obviously with axons is that as you said, we have like these inhibiting molecules, and partly it's because of the body not wanting regeneration in a way that links one part of the axon to a soma that is not its own. So as you said, you could get, for example, if you got one that is like a nociceptor, something like that, to another soma that is really important or is always firing up or something,
Yeah.
that could lead to chronic pain, for example. So how it is ensured that this regeneration that takes place, it's done like orderly and correctly, and is there some specific way, or is it just some techniques that are working better than others?
That is the 7 million, 10 million
Question.
pound question. Exactly. Right now, that is the holy grail. So I mentioned that there are groups around the world that can actually make actual de facto regeneration. You can see the axons growing. So there's, for example, a lot of people talked a lot about stem cell research in the noughties, and there's several groups actually showing that actually you can do quite a bit, and you can see a humongous amount of regeneration through the lesion, beyond the lesion, but nothing happens. The animal doesn't recover function. And how to guide it molecularly is completely unknown at the moment. We as a scientific field don't understand all of the little details that are necessary to achieve that. That's why I'm saying activity is one of the most potent ones. So if you're saying to the system, look, I need to do this with my arm, flex, extend. So how do I do this? I need to have this connection going over and over and over to that place. So you activate that same muscle. So there's signals that go back to the spinal cord saying, okay, that pathway, we'll call it pathway, this connection, right, is really important. Keep doing something to it. So in a philosophical sense, that's what we're thinking, that the signals that are coming from the exercise, the activity, so putting people on the treadmill, making people stand up, bear weight on their feet and try to keep a postural control and so forth, those signals are going back to the spinal cord and trying to guide this regeneration, so mold the regeneration. It's very much like if you think about babies trying to learn to walk, people trying to become super good athletes, or piano players or ballerinas or whatever that requires a lot of skill. You have to practice over and over and over. And with practice, there are changes that happen in the central nervous system. And this is what we're trying to recapitulate in this sense. Right?
So going back to these connections we were talking about, if people are studying like those same, like as you said, dancers, just athletes and all that, and how the central nervous system changes, could that help in your own research?
Yes,
in a way?
Absolutely. So one of the things that I like to remind researchers in the field is that unfortunately we don't understand how the intact system works. We don't understand how is it that a signal from up here in the cortex goes to maybe somewhere else in the central nervous system, basal ganglia, the brainstem areas, and then it comes down to the spinal cord, and you're able to do a triple back with a full twist. No idea how that happens. We have some idea. Sorry, we have some idea. It's not like we don't have any idea, but we don't know all of the details, and then those details become so important. As we know because when something goes wrong, like when you have a stroke, when you have a spinal cord injury, wow, it's difficult to repair because we don't even know how the intact system is actually working.
Now that we're talking...
So understanding how to improve function, so like a piano player, or a pianist I should say, a real skilled person, a real skilled dancer and so forth. Yes, understanding how that's achieved in an intact system will also help in other conditions like spinal cord injuries or stroke.
Talking about that basic science, or like how things work overall, I wanted to ask you, because even though the use of mice is widespread in research due to practical and ethical reasons, it is also true that these model species differ in many ways from humans. For example, their locomotion system is way different. I assume that this is also the case when it comes to neural connection maps in the two species. How do you translate what you see or induce in mice to human systems and physiology?
Yes, that's a really important question. It's a really important question in so many levels. So nobody actually wants to do research in animals. We do it because right now, to achieve the goals that we have there's no other there's no other way for us to do it. We cannot put everything on a computer model because we don't understand all the variables. We cannot just use cells because we need the system, the intact system, to, for example, see if the mouse or the rat can walk again. You cannot see cells walking that easily. So we need to use these animal models at the moment. So based on evolution theory, right, we know that there's conservation of functions amongst living beings, and because we're using a mammal system, there are lots and lots and lots of similarities. So even down to the genetic levels of control. So the system is primarily organized similarly in a mammal, right? The details aren't, and those details are quite important, as I mentioned before. So you need to be aware that the control of the hands, for example, in a mouse or in a rat, very different from ours, and our control of hands is very different from pretty much all of the other animals on this planet. So we need to take those things into consideration. But because that we're this stage of knowledge that we know a little bit, we don't know a lot. So this basic commonalities between species, we can target those and we focus on those first. So when we find something that is interesting and oh look, this might actually work, we need to move on to a different model. You almost never you go from a rodent model straight to humans. So in the case of spinal cord injuries, for example, it's very important to actually understand how you're going to perform the interventions. So maybe the surgery is different. You cannot transfer the tools that you use in a mouse to a human. So right now, what people have been doing is doing it in a pig model first. So it's a bigger model. So, the size, the anatomy is a little bit more compatible with humans. And if it's a pharmacological or a little bit more invasive model, they still use primate models before they go into humans to actually test if it's a cervical lesion, for example, that affects the hands. It's quite different in a mouse and a human, but it's not that different between a primate and a human. So you still have to have those steps in between.
I see. I absolutely didn't know that. I thought it might be the case, but I just didn't know. It reminds me, it's a bit silly, but it reminds me of this class I had with a teacher, and we were studying prions and some neurodegenerative diseases, and he said, well, in rodents, in some cases, they still function and it is fine. And he said, but obviously we cannot just go to the rat and tell them, do this equation or something to make sure they are properly functioning. And I think it's just that. Obviously, there's a huge difference, but as long as we can do something about it.
I'll tell you something interesting. You can also go the other way. So there's some experiments, actual experiments are more easily done in humans. Like you said, if you need to do equations, humans are better at doing that. So if you need that for your output, humans. So for example, I mentioned that the technique that I applied in rats is now being used in humans. The humans that have been able to walk again with epidural stimulation, you need to tell them, really focus on the movement. Think about it. Imagine it. Imagine your legs moving. You cannot do that to a rat.
That's true.
So, you can do even more thing with a human. And it's different. Obviously, it depends on what your interventions you're trying to apply.
So we've reached our last question.
All
And
right,
now
so
that-
quick.
Yes. And now that we are talking about humans, another thing that I actually picked up from your classes and I found really interesting is that their only concern is not just walking,
Yes.
and they have many other things they worry about. So spinal cord injuries not only affect the connection to the main muscles that help us move our legs or the limbs, but many other neurons. Could the studies that you're developing at your lab also help with sensory recovery or control of other many important muscles that we don't think about, like sphincters, for example?
Mhm. That's a really, really good question. Thank you, Marta, for asking that, because it's one of the things that if you don't have any knowledge about spinal cord injuries, you don't know that those things happen. So paralysis is just one of the problems that happen after spinal cord injury. So as you mentioned, depending on the level of the injury, other functions are going to be affected. Pretty much in every single type of spinal cord injury, your bladder, your bowel, and your sexual function are going to be affected. But depending on how high it is, other things will also be affected, like your heart, your blood pressure, your sweating, right? And all of these other functions that we don't even think about, and you can't really see because the person is sitting there in the spinal cord in a wheelchair, and you don't really see that. Yeah, so the holy grail is to actually find an intervention that will take care of all of that. And that means if you find a way of fixing the original problem itself, everything else should be fixable, right? So in the case of epidural stimulation, interestingly, even if you're just targeting the recovery of movement, we are seeing also recovery of bladder function, bowel function, some sexual function, sweating, and the other functions that I mentioned. And now there's a whole new wave of studies looking at control of blood pressure. This is one of the really important things in spinal cord injuries. And control of bladder. Even my lab now, I'm not just studying recovery of locomotion, I'm also studying recovery of bladder function, because if you don't have a spinal cord injury, if you don't know about it, you need to manage your bladder daily, hourly.
It just sounds
Right?
awful. Like,
Exactly.
It's just something that happens. It just happens. You
Exactly.
don't think about it.
Yes. And you can control it, you can wait, and you can do all sorts of things. But if you don't have the control, all of that is lost. And the quality of life, if you think about it, is quite poor if you can't have that kind of control. So if you ask, I think I mentioned that in my lectures also, that if you ask people with a spinal cord injury which function they want recovered, they don't say walking as their first choice. They always say some of these autonomic functions, bladder, sexual function, and so forth. Right?
Again, it's something we just take for granted.
Exactly.
But if we just got rid of them in our lives, it would be such a problem.
Yes.
So I really appreciate it. I mean, I don't know anyone that suffers from that, but as you said, many people suffer from that. So we should be grateful to you and everyone that is studying that, because you're just improving people's lives. And
Thank you. That's very kind of you
It's amazing.
to say. But it's our passion as scientists. It's a nice. I have to say it's not. I don't have to say it, but it's a real nice thing to see something that you started in the lab having actual effect on people's lives. That is really, really nice to see, right?
This is all for today. So thank you very much, Professor Ichiyama, for being here with us.
Thank you. My pleasure.
And thank you to all our listeners for tuning in today for another episode of Inno Podcast. See you all next time. Adiós.
Bye. Thank you.
Síguenos en las redes sociales @innocampus.uv.